All right. Well, pleasure to meet everybody. Thank you very much for having me. I'm going to have one of my colleagues, Josh, on the line here shortly. Since my screen sharing, you'll have to let me know when he jumps on. My name is David Moss. I'm one of the co-founders of INmune Bio. The idea of INmune kind of started around 2010, believe it or not. And it really didn't come to fruition in terms of an actual company in 2015. And the idea when we formed the company was really to focus on what we call the innate immune system. Most of the most of you know that there's two sides of the immune system. You have the adaptive and the innate, and they work together. Most biotech developments happened on the adaptive side of the immune system. Those are things like T cells, CAR Ts, checkpoint inhibitors, most monoclonal antibodies, et cetera, et cetera. But a lot less has happened on the innate side of the immune system. And part of the reason why is because on the adaptive side, it's very easy to do animal studies. And often research starts in a lab at a university with animals, and they're able to do their work with a drug, publish it, and then they try and translate it to man. On the innate side of the immune system, you can't do that. It's too difficult to do. And so you're really just left with coming up with an idea and then going right to man, which is more difficult. So we felt that there was a lack of drugs in the innate side of the immune system. And we also knew that the innate side of the immune system was kind of the first line of defense. And to really make a difference in disease, you kind of have to have the adaptive and the innate working together. Otherwise, if you just have the adaptive working, it's kind of like sending a boxer into a boxing match and tying one hand behind their back. You'll get some punches in, but it's difficult to win the fight. We also know that more than 90% of the animals on this planet do not have an adaptive immune system. They have just an innate immune system, and they're able to deal with many of the same diseases we deal with, cancer and infection, etc. And they do it just with their innate immune system. So we built the company with the idea that we were going to develop drugs that were going to fix, repair, and modulate the innate immune system. And that's why we call ourselves INmune Bio. That's what the I and the N is for. It's really for the innate side of the immune system. So we focus on immunology and inflammation. We have two platforms that do this. Both are in the clinic. Um, Here we go. And where we are in the clinic today is that our flagship program that most of us know us for is called XPro. It has about 80 publications on our website in many different disease categories. And it's a drug like nothing I've ever seen. It's a drug that's able to target what we call chronic inflammation or neuroinflammation and do it without any consequences. Um, there are many drugs out there that target inflammation. You've heard of steroids, you've heard of JAK inhibitors, TNF inhibitors, DMARDs, anti-IL cytokines, for example. But all of those drugs have serious side effects. All of them are highly immunosuppressive, and many of them cause other complications beyond immunosuppression. And XPro is the only drug I've ever seen that is potent enough to target inflammation, but not cause immunosuppression. Actually, it's immunosupportive and not cause other problems like neurologic problems, for example. And it means that we could use it in categories that have never been tried before. One of the beauties of targeting inflammation is that it affects the immune system. And unfortunately, what happens with age, and we all age because we can't stop age, can't intervene in aging, is as we get older, our inflammation increases. And it has nothing to do with really diet, exercise. Those are all contributors to it, but it really has to do with genetics and age. That's the vast majority of it. And as inflammation happens, wherever you have it, you have cell death. And that's something we want to slow down. Think to yourself about an elderly person. What happens? Their eyesight weakens, their muscles deteriorate, they start having memory problems. Those are all really inflammatory effects that cause cell death. We call that, another term for that is called inflammaging. And so XPro is a drug that's very unique. It's unique how it works, and it's unique in its therapeutic applications. We like to actually call it a pharmaceutical company in a bottle because it has applications well beyond what we're currently doing today. And that's what you can read about on some of the 80 publications on our website. But currently, this drug is in a trial in Alzheimer's disease, and it's gonna answer the pivotal question that if you properly treat neuroinflammation, what does it do to cognition? Our belief is that if you get rid of neuroinflammation without immunosuppression, the immune cells of the brain start to function again, and they start to remove debris, they start to repair the synapses and the neurons, and we'll talk about this a little later, and your brain health gets better. And if your brain health gets better, your cognition should get better. So this trial, which is currently running, it'll be fully enrolled very shortly in the middle of this year. And 6 months from the last patient in, we're going to have the answer of if you properly treat neuroinflammation, what does that mean for cognition? We believe that it will stop the cognitive decline, and some patients may actually cognitively improve. And I'm going to share with you some data from our phase 1 study that shows how that mechanism of action works and how we think about Alzheimer's. The second platform, which I'm not sure we're going to have time to discuss today, is also very interesting. It's on the innate side of the immune system, and it's around what are called natural killer cells. Natural killer cells are to the innate immune system what T cells are to the adaptive immune system. Natural killer cells are actually our first line of defense when it comes to virally infected cells or cancer cells. They do something called immune surveillance. Every day they knock off cancerous cells in our body. It's the reason we don't develop cancer. It's not the T cell. And it's a failure of the NK cell to not target these precancerous cells, which is the reason why cancer develops in the first place. And what INmune does is it changes what we call the phenotype of the NK cell. It changes it from a resting state, and it upregulates a whole series of proteins on the cell surface of the NK cell to change it from a resting state to what we call a memory-like phenotype, or a memory-like NK cell, which is now no longer resting, it's activated, and it has the ability to identify cancerous cells that it couldn't identify before, and it has the persistence to be able to kill them, survive, and kill again. And it also can do it in what we call, it's called a hypoxic tumor microenvironment, or let's just say a very hostile environment that the tumors create. It's currently in a phase 1/2 prostate program. It's in what's called metastatic castration-resistant prostate cancer. We have a lot of data that shows that there's a lot of resting NK cells in prostate tumor. There are not many T cells in prostate tumor. And if we can change these resting NK cells to a memory-like state, we should be able to reduce tumor burden and increase survival. And we'll have some of that data in the phase 1 program towards the end of this year, and the phase 2 program will be towards the end of next year. If that program is successful, we'll be able to go to the FDA and then apply for what we call a registration study, which means that we'll be able to run one more study after that, and if successful, that would be approval of the product. So, both of these are very unique programs. We feel that we're kind of writing our own playbook in the industry. If you look at Alzheimer's, we recognize that there's been a lot of failures, and we kind of understand the reason why there's been a lot of failures. There's been a lot of failures because we've been doing the same thing over and over again. Notice I didn't mention the word amyloid plaques or taus. That's where almost all the research and money has been. And we know what the results are. They're just not that great. We believe that Alzheimer's is really driven more by neuroinflammation and immunologic dysfunction of the brain. And we're the only ones to really take this approach. And if we're innovative enough and we're correct, it'll change research across all of CNS disease. With regards to cancer and INmune, we're using also an approach that's very different. Most people are trying to isolate an NK cell and engineer it and grow it and then give it back to the patient. That's complicated, expensive, doesn't necessarily work too well. We believe that all you need to do is just change the phenotype of the patient's own NK cells. There's a lot of history about being able to do that with cytokines, and we think that if we can do it, it'll be also a novel approach, which isn't just specific to a certain type of cancer either. It'll be able to lend itself to other forms of cancer. We have great data in, for example, like ovarian cancer and renal cell cancer and head and neck cancer and so on and so on. So if we're successful in prostate cancer, that'll be just the start. So so Florian, if that's kind of an overview. If you want, I can go into more detail on kind of milestones in the Alzheimer's program specific, or if you want to do question and answer, and then I can just pull up slides on the answer. I'm going to leave it up to you.
No, I think a few milestones, if you were to go through them, I think that would be great.
Super. Okay. So as I mentioned earlier, the phase 2 program in Alzheimer's will enroll the middle of this year, so we're not too far from that. And 6 months from the last patient enrolled, I don't know exactly what date that's going to be, we'll have the top line data. That top line data will answer the question about cognition. It'll probably take 2 to 3 months to get the secondary endpoints, just because there's a lot of work of compiling biomarkers and imaging. And once we have the full phase 2 package, assuming it's successful, we're going to have a meeting with the FDA around the middle of next year. And that agreement will come to an agreement with the FDA of what a registration trial looks like. We will apply for some accelerated approval pathways, which will allow us to potentially accelerate the approval of this program. And that approval can happen in 2 different ways. The first way could be that, let's say the data was just absolutely stellar in the phase 2 program. There's a chance, and I would say it's a low probability, but it's still a chance that we could get potentially conditional approval based on the phase 2 data and running then a concurrent phase 3 for confirmation. The more likely outcome, though, is that the phase 2 is successful and the FDA says, okay, hey, go do this now in a larger phase 3 study. That means that we only have to do one phase 3 study to approval. My guess is that the phase 3, if we do it, will start the second half towards the end of 25. And it'll probably be complete in 27. We do have some preclinical work. We think that XPro together with anti-amyloid will make anti-amyloid better and safer. One of the side effects of anti-amyloid is with an effect called ARIA, which is an inflammatory effect of the brain. This is more of a partnering strategy. We think XPro is way better than anti-amyloid. But the companies that have anti-amyloid drugs are big companies. And so we think this is more of just kind of interesting partnering bait to solve one of their problems and maybe squeak XPro in as a better drug. And then we have a second phase 2 program that's half funded by the NIH. It'll start the second half of this year. It is a treatment-resistant depression program. I will say that the number of people that have treatment-resistant depression is very, very large, unfortunately. So both the AD program and the TRD program are, both gigantic programs when you look at the patient populations, and they probably will both have price points that are very close to one another. You're talking many millions of patients in each of those categories. Once we're successful in the AD program, I believe the company will have a lot more resources, and we have a lot of interest in other CNS diseases like Parkinson's and MS and ALS and so on. So we will expand the XPro franchise. At that point. Over to INmune. We've completed the cohort 1 in the phase 1 program. We've already announced that. We've got the patients selected for cohort 2 will be enrolled shortly this month. Once cohort 2 is complete, we'll complete cohort 3. The whole phase 1 should be done around the third quarter of this year, and we'll be able to share some of the phase 1 data, I think, in probably, I'm going to guess Q4 of this year. I was going to say Q3, but probably Q4 of this year. That will then enroll into the phase 2 portion, and that will not be complete until the second half of next year. As you can see in the last bullet point there, once we have the full phase 1, phase 2, if successful, we'll have another end of phase 2 meeting with the FDA and determine what a registration study looks like for the prostate program. So those are the milestones.
All right, that was great. Thank you very much. You mentioned resources. So can you talk a little bit about the game plan when phase 2 results are out? So maybe let's think about a few scenarios. What are you planning to do if the results are disappointing? If the results are great? Or is there maybe also a scenario where maybe cognition is not really that significantly better, but you have a lot of positive evidence from biomarkers, things like that? So a little bit middle of the road, not really great, but not a failure either. So those 3 scenarios, can you talk a little bit about that?
Yeah, so let's talk about the failure first. So let's say we do this and we get an absolute bagel, we get 0, we get nothing, right? Which would be shocking to all of us. We're going to go through and understand what happened and why. It probably affects what happens with our TRD study, most likely, because there is a little bit of interrelationship between treating neuroinflammation. We do know from the phase 1 study and from all the 80 publications that we have on our website that it's a very durable drug. It absolutely reduces neuroinflammation without question. I can tell you with 100% certainty that we have a big reduction in neuroinflammation. If it's a failure, it's going to be pretty shocking to us because it probably means I don't know what it would mean. The drug's not interacting, not working. I don't know. We'd have to look at why. That's going to be a difficult scenario for the company. I mean, it would probably be a restructuring of the company, in all honesty, unless INmune is successful. If the drug is successful, and let me define what I think success is. If you look at I'm going to pull this chart up. If you look at the anti-amyloid drugs, their results are marginal at best. You get about a 30% change in the slope of decline, but you still decline. So you can remove all the amyloid, but at the end of the day, you still decline. Here, I'm going to share this. And so success to us, here we go. So this orange line and this solid blue line is the actual data of lecanemab. They actually had a statistical separation at 6 months, but this delta is really about 4 to 5, maybe 6 months of difference in terms of your cognitive decline. Patients that get neuroinflammation decline more rapidly than the average Alzheimer's patients. Patients that have, elevated levels of neuroinflammation decline more rapidly. In other words, their cell death is accelerated. And if you look at what success is to us, is all we really need to do is match this orange line, which wouldn't be a great result to us, but it's a great result for Wall Street because these drugs come with a lot of serious side effects, complications. It also limits the patient population they can go after because of the ARIA effects. Our drug doesn't require all this MRI imaging and all this, complicated infusion. It's something that could be administered at home. So success would be from here to here. We think that most of our patients are going to do well better than this orange line. I think some will be stable, some will probably be in between the orange and the green. And we actually think that some may actually improve above this green line. If that occurs, I mean, I think what's going to happen is that it's going to change the way Alzheimer's, dementia, and CNS research is looked at. Because for the first time, um, there is evidence that neuroinflammation, if treated properly, can make a difference in a disease, and that will cut across pretty much almost all CNS-related diseases. Now, if the success is marginal, let's say that, what would marginal be? Let's say it's marginal would probably be equal to lecanemab or worse. I think what we'll do is we'll again, we're going to sift through the data, try and understand what happened. And we're going to be honest with you and our shareholders and ourselves. We're going to say what it is. And we're not here to play games and try and cherry pick data that shows something positive to try and keep a company alive. We're here to really try and make a difference in a disease. And we're betting along with our shareholders, our employees, and our patients that we're treating that we're going down the right path. And so we either go down with the ship, sadly enough, or we're gonna make a difference in the disease. So, I mean, best I could answer, Florian. And Josh, I don't know if you have. I want Josh to introduce himself, because Josh is he does a lot of our corporate work, and most importantly, he's a patent attorney. He's somebody who actually helped write some of the original patents that the company has, and had his own practice and joined the company. So I brought Josh on so that some of you can meet some of the other people at the company, but also I'd like him to give his perspective.
Sure, David. So yeah, I'm Josh Schoonover. I'm general counsel for INmune Bio. But in addition to contracts and transactional matters, I actually specialize in patent law. So it's somewhat unique for a small biopharma company such as INmune Bio to have an in-house patent lawyer. What that means, and I think what it showcases to investors, is just how seriously we take our patent position, our intellectual property portfolio. We have someone, namely myself, who spends all day long thinking about these things and taking action to position our company with strong intellectual property. So I think that's pretty much the sum of what I do in my day-to-day activities. I guess if you'd like, I can talk briefly about our intellectual property.
I
Yeah,
think we
please
get.
do.
Yeah, let's do that. And then if you have an opinion also about the data and the readout and your reason for joining the company, I think that'd be helpful too, Josh.
Yeah, so I had my own patent law practice in San Diego, and I joined the company after becoming involved and really prosecuting the intellectual property, reading all of the prior art documents that are out there, learning about TNF, and in particular, what INmune Bio does, which is selectively targets a form of TNF called soluble TNF. It's different than what's called transmembrane TNF. And the reason this is important is it goes back to some of what David mentioned earlier about some of the complications with other TNF inhibitors. What XPro is really is the next generation TNF inhibitor. It has all of the beneficial aspects of a TNF inhibitor, but it has, in addition to that, it's not immunosuppressive. It doesn't cause demyelination of neurons and some of those things David had mentioned. So it's really a very, if I can say it this way, it's a very sexy drug. It was really designed with modern technologies, and it selectively targets that bad form of TNF without interfering with the good TNF. So that's what piqued my interest. And knowing traditionally what TNF inhibitors have done in the commercial marketplace, I knew, all of those are blockbusters, Humira, et cetera, Enbrel, et cetera. And knowing that we have a drug that is the next generation, then it really opens up a whole series of indications that those drugs cannot target. Those are the neurological conditions, things like Alzheimer's disease, like we're looking at now, and even more. So that really piqued my interest, and I thought there was an opportunity to join INmune Bio, really help to pursue some of those things and develop the intellectual property portfolio. But also, I just have, I got to say, I drank the Kool-Aid and really believe in what this drug can do, and really excited to see where we can take it. So that's sort of my background and my opinion on this. And I really can't emphasize enough that graph that David just pulled up, because what it really shows is one of Biogen's drugs and the sort of the delta between placebo group and the effect that they showed. Now, we know their effect wasn't super significant, but that effect that they have in comparing what our placebo group looks like, meaning those inflamed patients, our placebo group will decline much more rapidly than the placebo group in the lecanemab trial did, which means the effect that we'll see potentially can be significantly greater, because we know what inflamed patients look like and how their disease progresses. And I think if you can just recall that graph, the delta is significantly much more better. And it allows us to do a trial in 6 months as opposed to 18, which I think can be, there's a lot to say about that. One I'll just point out quickly is, patients who have Alzheimer's disease deserve a chance. And to put a patient like that on a placebo for 18 months is really, in my mind, not fair to that patient. So when you have an opportunity to do clinical trials that are, for example, 6 months, such as XPro, I think it's really a much better option for those patients. And really, at the end of the day, that's what we're all about here at INmune Bio, is we're trying to develop treatments that make a difference in patients' lives. So I'll stop there, and we can return back to the conversation. I hope that was enough. You can tell I'm very spirited about the company, and I hope it shows.
Well, I think anybody who follows the company knows that the whole management team is really all in, not only with the money, but also with the spirit. So I think that is certainly great. I don't know if you have a good answer to this question, but I'm not really aware of a sort of bearish case on the company, and yet the company seems to be more or less ignored by a lot of investors, certainly if I look at the market cap. And also, if I look at the 1.7 million shares sold short, I have no explanation for it. I would have had an explanation before you raising money. Now, also with the Russell inclusion and with you entering a phase 2 trial with certainly not bad prospects, I'm not sure where that is coming from, especially since there seems not to be a lot of controversy. So I don't know. What are your sort of thoughts about that?
I think, I know as much as you do, to be honest with you. Everyone I talk to about short interest in their company or professionals in the space, everyone's got a different answer. Nobody really knows at the end of the day. What I can tell you, though, is that I believe, and this is my own personal opinion, that the way we did the financing, the two different steps, one was obviously insider led with 20% of our own money coming in. Josh was a big participant, by the way. Thank you, Josh. And then followed up by a single investor who just was tired of waiting. And now that we're funded well through the clinical data and we're going into the Russell, I feel like we've trapped those guys. I feel like we have a chance to really give it to them. So I don't know where it comes from. I think it's just basket shorting, where a fund says, hey, look, I don't like these money losing biotech companies, take 200 of them, 300 of them, 400 of them. And if they fit some sort of algorithmic mechanism, just short them, and they have no idea who we are. I think it's just we're in a basket. That's my guess. I could be wrong. I would say that the reason we're unknown is that it's really hard to be an innovator. Somebody who comes up with a new idea and then goes to try and drive it, they're literally crazy until the minute they prove it. And and then they become geniuses. I'm not saying we're geniuses. I'm just saying that they're considered crazy before. So biotech space is just not that innovative. If you look at all these large, sophisticated, well-known funds that fund large biotech companies, they're really funding a playbook. They're saying, oh, this is another monoclonal antibody or another ADC or another, small molecule that hits this target a little bit better. It's 10, 20, 30, 40, 50% better than the product that's on the market. And because we know that large pharma already has products in this space, if we get a little clinical data, they'll buy us, right? And if you look at what we're doing, is we're targeting Alzheimer's disease in a completely different way. We're using a drug called a dominant negative protein that's no one's ever heard of. Have you guys ever heard of a dominant negative technology before? Probably not. And if we're successful, there'll be other dominant negative technologies behind us. Look at the first CAR T companies, Juno and Kite, especially Kite. Kite, the only reason I say Kite is because it didn't have arch venture partners behind it like Juno did, which gives it that kind of aura. But Kite, was struggling as a company for a long period of time. I think they were an OTC company before they became NASDAQ company, and they struggled until they had clinical data, and then they got approved. And now there's a couple hundred CAR T companies out there. Before there were only 2. And Kite was the first one really that struggled. So when you're an innovator blazing a new trail, we're writing our own playbook. And until that playbook is accepted by industry, I don't think we're going to get the market cap or the love that we should. And that venture return, therefore, is still left in the marketplace. The same thing goes for INmune. All of the other NK cell companies are sourcing an NK cell from something. And by the way, they haven't even proven that they're working well, so they don't have good values. And then they're using cytokines to try and boost their NK cell and change its phenotype, and it's still not working very well. And they have a very expensive therapy to deliver that they might have to give multiple times. It's not like a CAR T where you give it once and you're done and you can charge $400,000 for it. They have a therapy that costs a lot to make, and they might have to give it multiple times, which means it's not even commercially really makes sense. And so, we're not another monoclonal antibody. We're not another small molecule. We're not another T-cell engager or TIL or CAR T. We're blazing our path there. And, what excites me as someone who's been in the biotech industry for a long time is I don't really want to be a Me Too company. I want to try and do something different that has a chance to really, truly make a real change in a disease. You guys, I don't know if you guys remember like the original Apple computer commercial when they're competing against IBM and Microsoft and all this, and they have that lady that's running down the aisle and she's throwing the sledgehammer at the screen trying to break the mold and the psyche. That's the way I feel with INmune Bios, that we're out there, we're the underdog, we're taking a new approach, and we really are trying to do something very different. I mean, look at our trial. There's so many people out there that says, oh, if you're not doing 18 months in Alzheimer's, you don't know what you're doing. And you know what? I say the opposite. If you're doing an 18-month trial, you don't know what you're doing because you're guessing, and you need to run 18 months in a lot of patients because you have no idea how your drug works. We know how our drug works. We know what it does. And that means we can choose our patients better. We can choose our biomarkers better. And we can shorten our trial and measure those biomarkers, and you get a better chance of success, and you don't have to run these long, large trials. So what we're doing is just not what the industry is used to, but if you want to make change, that's sometimes what you have to do.
I think that was a really, really good answer, which leads me to a natural follow-up, which is you are confident that if the trial works as you anticipate it to work, that recognition will be there from other pharma companies, from the market? Or do you think there's still a possibility that people are going to say, oh, well, they got lucky or something like that?
I mean, they can say whatever they want, but when you run a gold standard trial, that's what the industry runs. It's a blinded, randomized, placebo-controlled trial that's going to have a p-value, and it shows what it shows. I mean, how do you argue with that? I mean, it's like a mathematical equation. I mean, either it comes up with a number. I mean, I think like even though we have it only, it's open label, it's our phase 1 study, it's not a lot of patients. I mean, we did it during COVID. It was only 3 months, and we had some patients try and stay on as long as they could, and we still have 2 people that are demanding the drug today. Even though it's a small N, to me, that's like really cool. You've changed 2 people's lives, right? I mean, it like when I see that video, it honestly brings tears to my eyes.
I would agree. I would agree. Speaking of open label, can you talk a little bit more about your decision to end the extension trial?
So the extension trial was going to cost a lot of money. It was going to cost, around $10 million to run. We were getting a relatively high enrollment rate, and it was using a lot of drug. It was going to use a lot of drug. If we kept it up, it would have been a big expense to the company, and we would have had to go do manufacturing earlier than we expected to. And the manufacturing is probably somewhere in the $7 to $9 million range to make drug, just so that we could have drug for the phase 3 to do the phase 3 on time. And because the trial's enrolling at a reasonable pace, we're going to get data from the phase 2 blinded randomized trial pretty soon. And I don't even know if we get open label data by the time we'd have the phase 2 data. The other thing about the open label you have to understand is that when patients go into the open label, it is an open label study, just like the phase 1. So are people going to look at the data just like they did with the phase 1? Probably. And we don't know if those patients are on drug or off drug or on placebo, I'm sorry, when they enter the trial. We also don't sample as often in the open label as we do in the trial. And that's by design because it's expensive every time you do a sample, which is a cognitive test or an MRI image, which means that the patients have to go out, enough patients have to go out longer for us to get a reasonable amount of data, and we're not there yet. So it just made, financial sense to close it. I would say that if our market cap were higher and I had an extra $30 million in the bank, we probably wouldn't have done it. But that's not the reality of the marketplace. It's not where we want to dilute ourselves. And at the end of the day, we're at the point where people just want to know cognition. They want to know, they don't, these MRI images and these CSF images and this proteome and all this cool, neat, points in the right direction. But what does everyone asks us, whether it's a sophisticated institutional investor or or an average or just a retail investor, what happens with cognition? And we're going to get that answer real soon here. It's what the company's. It's the most pivotal time in the company's history, is the readout of that trial, and we're close to it. So it from a financial sense, and from a drug use sense, and from a data reporting sense, it was too much of a burden.
Very good. Speaking of financials, one point I wanted to highlight is, as long as I've known the company, you have done a remarkably good job when it came to raising money, always from a position of strength, never really, always in a way that benefited the company, and not letting yourself be trapped. And so I have to say, compared to pretty much every other biotech company I know, you have done a remarkably good job. That's just statement from me. And I
Thank
have
you.
a last question, and then maybe someone from the audience, which is your next financing. Do you anticipate that to be an equity financing, or do you think you will make use of a partnership or some collaboration with Big Pharma? What are your thoughts about that?
So first of all, thank you very much for your comments. I've always said this to everyone that we speak to that asks the question about dilution, because they get, they're so conditioned by, as am I, by how they've been treated by other biotech investments that they've made. I think it's pretty unusual where you run across a company that has 3 management members that are founders that still own a lot of stock and have raised a lot of money, right? I'm as sensitive to dilution as anybody, as is RJ, as is Mark, as is Josh, as are our employees, right? We're as sensitive to anybody because we believe in what we're doing, and we want to covet our equity as much as we can. We know there's going to be dilution. And if there's dilution, we want to try and do it as smart as possible, but it's going to happen, right? The thing that we always ask ourselves is, if we get to the end game, does this dilution that we take today matter? It does matter, but I think it matters a lot less if we're able to ring the bell on what we're trying to do. So that's the dilution question, but thank you for your comments, and we continue to try and manage it as best we can. It's not an easy feat. And by the way, we like to say that there's 2 ways you dilute the company. You either dilute it through equity and capital, or you dilute it through assets. We've had the chance to dilute our asset, but we didn't want to because it's just too early. If we dilute our asset through a partnership or a sale of an asset, we want to do it right. So we're close enough to data where it's better for us to get data and then do a partnership. My belief is that if we report data that is far better than the anti-amyloid, and we maybe even have some patients improve or not, but it's let's say equivalent or better than the anti-amyloid, I think that every large pharma company that has an interest in CNS disease, in dementia, will be wanting to talk to us. And I think that partnerships with these companies don't happen quickly, but when you have quality data, I think there's going to be a little bit of competitive field out there. A lot of these guys know about us. They know what we're doing. We do talk to them on occasion. And I have a feeling that, things will heat up. At the end of the day, we see this as a valuable franchise to the patient, to our shareholders, and to ourselves. And we are operating as if we will fund our own phase 3 study. But if anything comes up that we think is valuable, that is properly structured with a reasonably agreeable price, we'll entertain it. But we have pretty big numbers in our head.
Awesome. Thank you. With that, we open for questions from the audience. Todd has 1 or 2 questions, I think. So Todd, let's go.
Thank you, Florian. And David, great to hear from you again. We've spoken several times.
Yeah, thank you, Todd.
I also want to just compliment you authentically about always being so exceptionally clear when you discuss anything about the company. And you seem to know more as a CFO, obviously a co-founder, about the science than almost any CFO probably knows about the business, especially science. So kudos to you, and thank you again for always being so clear. So I have a question about something that I'm not so clear about. I might have misunderstood you. And then I have a question for Josh, and I'm going to put them back to back, and you can answer them however you like. You said something about Parkinson's, MS, ALS, maybe other CNS diseases. Are you saying that assuming you ring the bell with phase 2 results for Alzheimer's and assuming you raise significant capital on the back of that, are you saying that you would expect to immediately, obviously immediately meaning not the next day, but go right ahead with initiating a phase 2 study for those other CNS diseases? That's the first. Is that, if there's a simple answer, then I'll wait for my question.
Yeah, no, we have a drug that remyelinates. Demyelination is the hallmark of MS. We have great data in Parkinson's. NFL might be an approvable endpoint for ALS, for example. We don't know that yet, but it seems that way. And there's been no real drugs. And we have great NFL data in at least AD patients. I don't know what it looked like in ALS patients, but I assume that their brains are relatively similar. So And we know that neuroinflammation cuts across a lot of diseases. There was Baird did a KOL webinar on us a couple months ago, and we had a KOL there. Her name is Dr. Malu Tanzi, and she talks about neuroinflammation cutting across a lot of CNS diseases. So I would encourage you to try and dig that up and listen to it. In fact, I think one of our old press releases that announces it has the link to to that KOL event, and it's probably still available. I haven't checked, but it probably is. But the beauty, sorry, last thing, Todd, and then I'll let you go. The beauty of XPro is that we're not just an Alzheimer's drug. Like an anti-amyloid drug is just Alzheimer's and they're done. I mean, they're done. That's all they have. And the drugs don't work that great. Alzheimer's is just the start for us. And by the way, I really like what's going on in DMD. Steroids are terrible for these kids, and yet that's the standard of care, but they're highly immunosuppressive and cause problems long term. They probably actually exacerbate the disease. If you can replace that with XPro, way better. And in fact, there's a lot of other disease categories, cardiac disease, a lot of ophthalmic diseases. We've got great stuff in ophthalmology that we really like. These are things we'd like to, pursue. Inflammation cuts across. I'll go back to my favorite statement. You should type this into Google because it comes from the WHO. More than 50% of deaths are attributable to an inflammatory disease. And there's never been a drug that can target inflammation without serious consequences until XPro, is our belief. And the way you treat cholesterol or the way you treat like statins for blood thinning is, you're treating as a class. I think you could treat inflammation just that same way, but chronic inflammation is not yet considered a disease category by the FDA. They're getting there, what they call inflammaging. But I think that, this is a drug that treats inflammaging, and frankly, that lends itself towards, many different things. So I'm sorry, Todd, long answer. Let you ask Josh the question.
No, that's fine. Thank you, David. Before I ask the Josh question, so are you saying that assuming you raise several $100 million after phase 2 results, I'm not asking you to confirm that amount, but let's just say you do. Does that open the door to beginning of phase 2 in multiple concurrent trials?
It does. It does. And I will say that these are not, because they're phase 2 studies. I mean, these are, $20, $30, $40 million trials. They're not expensive, but yes, it does.
Okay, great. Thank you. And Josh, question about XPro patent coverage. In the most recent investor presentation dated April, it said something like 2033. So first of all, is that only for Alzheimer's disease as an actual use, or is this composition of matter? And also, does INmune have exclusive rights, or is there some end date of exclusivity, and how does Xencor fit into all of that? I know that's multiple questions.
Yeah, sure. I think I can tackle all that, Todd. So thanks for the question. So the 2033 is based on, at the moment, I would say one of our key patents that's in licensed from Xencor. It's an exclusive license, and it covers use of XPro to treat neurological diseases. So this is more than just Alzheimer's. This is Parkinson's, ALS, you name it, all of the neurological conditions, even includes stroke. We define stroke as a neurologic disease in the context of that patent. So that's the 2033 coverage, but there's actually more because and that's a method of treatment patent. We have also composition patents. So for example, one of the things that occurred, as developing a drug goes, you learn things and things change. And we have found the XPro molecule that's made now basically has an open, we call it succinimide ring in the linker. And this provides an avenue to obtain composition of matter IP. So we have pending IP on what I would say is the bioconjugate of XPro. So it's not just the active protein, but also the PEG and linker that's attributed to it. And we have we have a series of other things, formulations, compositions, methods of treatment that cover the molecule. So I would say although today we can definitively say we have coverage through 2033. The next aspect to that is the patent term extension, which you may know the USPTO and other patent offices around the world will give up to 5 years of patent term extension day for day for clinical trial development. So let's say we spend 5 years in clinical trials, that takes us from 2033 to 2038. So it's somewhat of a shiftable mark. But we're currently at 2033, possibly 2038 with that extension. And then we have other IP that takes us well into the 2040s. We made an announcement, so I can say this as public information. We filed IP on a antibody, a anti-drug antibody assay that was custom developed. So things like that give our program a lot more protection and coverage, and again into the 2040s. And then another thing that we build into our exclusivity strategy is the reference biologic exclusivity. This is completely different from patent exclusivity. It's a regulatory exclusivity. Is essentially the FDA, and again in other jurisdictions they have a similar rule, but the FDA for the US example will not even accept a application for a molecule with the same or similar mechanism of action. And then there's another period of years where they won't approve it. Effectively, it's 12 years in the US. So there's 12 years of reference biologic exclusivity, which counts from the date of marketing authorization, which, let's say it's 2028, that would carry us 12 years to 2040. So the dates as far as exclusivity, again, to summarize, definitively 2033, likely to 2038 with the patent term extension, and into the 2040s with the what I would call the other patents related to the XPro program and also the reference biologic exclusivity. So the takeaway is we have a very strong IP position.
Okay, that's great. The only reason I was asking that is because in the investor presentation for the non-legal minded people, it looks like if XPro is approved, let's just say approved for Alzheimer's in 2028, let's say, or even 27, if the patent expires in 2033, despite all the things you said, if that's not clearly communicated to the investment community, that can be a concern that a lot of people have. You have this great drug.
Yeah.
Even if you get some marketing exclusivity from the FDA, it's like, well, just when the party's starting, you give it up. And that might be something unsolicited advice I would give would be, especially given your position in the company, to have a little more communication on, what you can really say is probable in terms of IP protection that go well beyond what looks like a kind of a short cliff.
Sure. Yes, I'll definitely take that into consideration for future revisions of that investor deck. I will say it is a fine line between what we can say when some of these things are speculative, as far as how many years the USPTO will extend a patent, or what the reference biologic date will be when the marketing authorization will be received. But that being said, I think there are notations that we can make to perhaps make that more clear. So thanks for that.
All right. Thank you very much, both of you.
Yeah, thanks, Todd.
Yep.
Ashley?
Hi. I've been following the story for a few years, and
Thanks, Ashley.
mostly through Dan and involved.
Yeah.
So I had a couple questions. One, sort of from the commentary on the last earnings call, it sounds like you guys are raring to go on the TRD study. It's a third funded from the NIH. Are you able to kind of move through that quite quickly? I noticed sort of no endpoint timing is sort of given. Do you sort of have a picture of how that enrolls, and whether you can move that to the end sort of before your sort of next funding?
No, good question. We like the TRD study a lot. It's not a large study in terms of number of patients. That study is going to, it's going to start towards the second half of this year, but we're not going to get that trial enrolled till probably the end of 25, realistically. So, I think what's going to be the big data point for the company is going to be this AD02 readout, and that's going to determine the next step. So the TRD study is going to be a secondary follow-up to that. We like it because there's already there's TNF trials that have been done in the past that kind of give us a guideline of how well it's going to work. And so it's kind of more of our assurance trial. The other thing that we like about it too is that if you look at the companies that are in the space like Sage and others, their drugs just aren't that great. And so we think that it can be a real kind of like what we're not as big as what we're doing in AD if we are successful, but it can really make a big change in the treatment-resistant depression space. So, but it's really secondary to the AD program in terms of value. Sorry, timelines.
I guess as you said earlier, if there's some links between the two kind of Alzheimer's and depression, that you kind of maybe have most of your answer in kind of the winter. Okay.
Yeah, no, you're right. And the other thing too, that the links are kind of interesting, right? Because patients that have Alzheimer's, they can get aggressive, they can get depressed, they don't sleep well. These are all neuroinflammatory drivers, right? So there is a link between some of the clinical observations you see with dementia patients and depression patients.
Okay. And the other question was maybe a little more general. It seems like over the past couple of years, sort of the scientific community has maybe woken up a little bit more to inflammation as it relates to Alzheimer's, and certainly seen more articles on it.
Yeah.
But I think you touched on a little bit why you guys are sort of maybe unloved by the investor market. But if you've got something that sounds like it could be revolutionary coming out sort of in the winter, if the results are good, how come it's sort of not on the front page of every scientific magazine right now? What's holding that back, I guess?
It will be if we ring the bell in the phase 2. They want to see robust data, right? And robust data to them is a blinded trial. So that's why you're not going to see it there. People know about it in the industry. I think what's really missing, and the most important thing, and I say this a lot recently, is there's a history of treating neuroinflammation that has not been successful. And the reason why is because either A, they weren't potent enough. In other words, they weren't going after the master cytokine or the pinnacle of inflammation. But the most important reason is that they're immunosuppressive if they were. And so I think what's being missed in the community and in the space is that it's equally important in how you treat the neuroinflammation than just treating the neuroinflammation. And even the big companies like Alector and Denali and some of the others that call themselves neuroinflammation companies are missing that point. And so does the general scientific community. The how is so important. Because what's going on is that we're relying on the immune system of the brain to do the job. And so we're really resetting the immune system of the brain by getting rid of neuroinflammation properly. And that's never been done before. And so until we blaze this path and do it, I don't think we're going to get onto the front page of, Science or any of this stuff. And then if we do it, I think, they'll be clamoring to have us on the front page. It should be the case. And then what's going to happen with the scientific experts? They're going to, oh yeah, I knew that was the case, is what they're going to say. Yeah, of course, treating neuroinflammation with immunosuppression. Yeah, that's the answer. Well, if that was the answer, why weren't you doing it 10 years ago, 20 years ago? A, they didn't have a tool, but B, they didn't think about it.
Great. I think that's a great answer to the question.
Well, no,
thank you very much. All the best.
No, appreciate it, Ashley, and thank you for your continued support. I know it's been a long, difficult road, but it's hanging in there is important. I want to emphasize, our company's mission and has really never changed, right? It's still the same company doing the same thing, trying to get to the same goal, and we're getting close to it finally. So I really believe we're going to be successful. I can't promise you that we will be. It is a biotech company after all, and we have all the risks that every biotech company has, which can very much equally be failure. But I think we've, done the best we can to try and manage the biology, look at the biology, look at the MOA, design the trial as best we could to give it a reasonable shot. And I'm kind of excited to find out what's going to happen. I'm going to find out, the same time you guys are. I'm not going to know the answer till maybe a day or two before you guys know. So, I'm as excited as if not more excited than anybody about this. But we really want to run this through to see if we can truly make this work, make a difference. And then our world changes, right? The whole world changes in terms of what we can do from a financial standpoint, a development standpoint, partnership standpoint, et cetera, et cetera. Yeah, somebody else has a hand up.
Yeah, Chris Tuttle.
Chris. Yeah, Chris.
Oh yeah. Two things. One, just wanted to share that it's been a little while, but we back tested a model when I was on the sell side of, do you go long, all biotechs soon to report clinical trial results, or short? And consistently, they're shorting all biotechs within a window of having clinical trial results, and now it's far outperformed going long. And it could, there's no narrative. It's just a back test, right? It's just numbers. But reflecting on the investment industry over the last however many decades, is investors tend to be optimistic and to project forward positive results, because that's sort of the nature of the markets. But the mathematics of it was, you'd be far better off shorting every single one. You'd make a lot more money. So that could be, consistent with your point about it could be algorithmic, but we'll never know.
I think you're right.
My question, though, is, you mentioned a couple of those other molecules which have been big successes, and I'm not an expert in drug manufacturability, but for some reason I sort of had the impression that those were not the easiest to manufacture drugs. So I just wanted to touch on your views of, if you're successful, your ability to scale up and have these drugs manufactured, whether or not there are any potential gotchas or roadblocks in you being able to take that commercialization phase and not have any hiccups there.
Yeah. Let me come back to that. I want to comment on your first observation, because it's really interesting. Most clinical trials fail. So there's a high percentage of failure, number one. And the more recent thing we're seeing is companies that have failed trials and then trying to put out press releases like they're successful, by picking and choosing what they want, which I think is really bad for the industry. The other thing too is that, you obviously look at, like, you mentioned about investors tend to be optimistic. You're absolutely right, because I always remember from business school, if you look at the premium on calls versus the premium on puts, the premium on calls is always higher because people are more optimistic and they're more likely to buy calls than puts. And therefore, it's good to sell calls if you like a covered call, because you get more of the premium. Josh, I'm going to let you answer the question first, and then I'm going to come back to it.
What was the question again about the manufacturability? Is that it?
Yeah.
Okay,
because you're closer to it.
Yeah, so I would say, we really have those two programs, right? So DNTNF, and then we have INmune. Let me start with INmune. So INmune, I recently went over this with Mark, and he's got a slide. I don't have it handy, or I would share it, but the takeaway is he's got it figured out to where a bioreactor the size of a human being produces enough to do a phase 3 trial. So I have no doubt we're going to be able to manufacture INmune and scale up as needed to develop it and then to commercialize it. DNTNF, we're making it in E. coli standard manufacturing done for biologic proteins. We do have sort of a multi-step. We have a conjugation with polyethylene glycol PEG, and we have a fill and finish step. But altogether, these are conventional processes that are done, you know, in many sites all over the world, and they do improve. They tend to improve as, you know, things do in time. They figure out new ways to, you know, get a higher yield on a protein, or to you know, speed up the process for conjugation. But I would say even with today's technology, scale up and manufacturability is not a problem for DNTNF at all.
I think the bigger question for us is going to be, you know, what does the data look like, and then how do you pencil that out with regards to demand and need? That's an unknown.
All right, great guys, that's very helpful.
Yeah, there are what are called CDMOs that actually you contract to manufacture, and that's what we'll do. I often joke that, you know, if we're successful, it might just make sense to build our own manufacturing facility so that all we do is manufacture X-Pro nonstop on our own, but that's a problem to figure out down the road.
And have you do you have CDMOs that you've worked with before, or that you feel you have something for?
Yeah, we work with one called KBI. They're well known, very established. We operate out of their Boulder facility, Boulder, Colorado. And you know, a lot of big pharma use them to manufacture commercial drugs and experimental drugs, things of that sort. These guys are pros. It's not cheap to do, because you go off and you'll do a run and you'll produce, you know, 5, 6, 7, 10,000 doses at a go, a single batch. And so it gets expensive. The input, the material, the time is expensive. On a per dose basis, it's not. But then when you run commercial grade material, you run this consistently. So you do repetitive batch production rather than single dose batch, like commercial scale, and it gets a lot cheaper because you do it on a continuous basis. And so it becomes repeatable and cheaper. And you know, you order your materials in bulk quantities, so it's a lot cheaper than one-offs and so on.
All right, great.
But manufacturing is something that we've got down on both programs in terms of they can be commercial scale, is what you need to know. And we've got them even at a clinical scale. They're at a price point that is already something where we have enough margins to do very well. And that margin will get just that much larger once we get to repeatable commercial production.
Awesome. We are over the hour already, and I think there are no more questions anyways. So
This is the first time I never got a question from Steve or Dan.
I'm shy.
Yeah, no, I get it. I think does it mean I finally answered all your questions, Steve?
I think he's
Nope,
just holding back.
Yeah. Well, listen, I always say this, our shareholders are our lifeblood. We know that. And without good shareholders, we can't make this business work. It's the shareholders that give us the price that allows us to raise capital that we can invest in our clinical programs. So we appreciate our shareholders. We're here to share and answer any question. We're investors like you. You know, so we understand what it's like. We try and run things prudently. We make mistakes too, but we try and correct them, and we do listen. We like negative feedback because that's how you learn. So if there's anything you don't like, share it with us because we'll try and improve it. And you know, I always like to say my favorite quote, and it's from Warren Buffett from ages ago. He doesn't say it anymore, but it's, you know, I think really good companies are built when investors think like management and management thinks like investors, right? And that's the way we try and operate. And you know, we're just trying to develop these drug products and try and make a difference at the bedside. And if you do, everything else follows.
Awesome. David, Josh, that was great. Thank you so much.
I appreciate
we
it.
appreciate it.
Likewise. Well, thanks everyone, and thank you for being a shareholder.
Absolutely.
Thank you.
All
Thank
right.
you.
Bye-bye.
Bye-bye.
Neither Breakout Investors nor any of its affiliates makes any representation or warranty, express or implied, as the accuracy or completeness of the statements or any information presented by this podcast, and any liability, including in respect to direct, indirect, or consequential loss or damage, therefore is expressly disclaimed. The views on this podcast expressed are those of the speaker and not Breakout Investors. No one on this podcast is an investment advisor, and no one is providing investment advice. Before investing in any company stock, you must do your own research. Thank you for listening.